In 1952, one photograph quietly changed biology forever, yet the woman who captured it never saw the Nobel Prize. Today, we rediscover Rosalind Franklin's remarkable, unfinished story. In 1952 London, Rosalind Franklin captured Photo 51, the clearest image of DNA. 10 years later, others accepted the 1962 Nobel Prize in Physiology or Medicine. Rosalind Franklin, the unsung architect of DNA. To understand why Photo 51 mattered so much, we need to step back into Rosalind Franklin's world and the science race of the early 1950s. Rosalind Franklin was born in London in 1920, a brilliant chemist who mastered X-ray crystallography long before arriving at King's College London in 1951. By 1952, DNA was known as life's genetic material, but its structure remained a mystery. Cracking it became a scientific race across London and Cambridge. At King's College London, Rosalind Franklin wasn't chasing glory. She was chasing accuracy, and that would make all the difference. In 1951, Rosalind Franklin joined King's College London to study DNA with X-ray crystallography, sharing the project and a tense working relationship with physicist Maurice Wilkins. Rosalind Franklin demanded rigor. While some guessed DNA's shape, she insisted X-ray crystallography patterns and mathematics must lead, even if that slowed dramatic theoretical leaps. Meanwhile, just up the road in Cambridge, another pair of scientists were taking a very different approach to DNA. At Cavendish Laboratory in Cambridge, James Watson and Francis Crick built physical DNA models, sometimes leaping ahead of data, racing to solve the double helix puzzle first. Back at King's, Rosalind Franklin and graduate student Raymond Gosling perfected techniques to coax DNA fibers into orderly forms, producing increasingly sharp X-ray crystallography images. Then, in early 1952, everything came together on a single piece of film. In 1952, Rosalind Franklin and Raymond Gosling captured Photo 51, an extraordinarily clear X-ray diffraction image revealing DNA's helical nature with striking, unmistakable symmetry. Without Rosalind Franklin's knowledge, Maurice Wilkins later showed Photo 51 to James Watson, giving Cavendish Laboratory a shortcut to crucial measurements about DNA's double helix. What Watson saw in that brief moment would turbocharge the Cambridge model building effort. Seeing Photo 51, James Watson later described realizing DNA must be a helix. Those clear X-ray crystallography reflections narrowed possibilities dramatically for the double helix structure. Armed with Rosalind Franklin's unwitting data, James Watson and Francis Crick quickly refined their DNA double helix model at Cavendish Laboratory, confident they'd finally cracked life's code. While celebration brewed in Cambridge, Rosalind Franklin was quietly finishing some of the most precise DNA measurements the world had ever seen. At King's College London, Rosalind Franklin independently reached key insights about DNA's double helix, carefully calculating dimensions that closely matched the model emerging at Cavendish Laboratory. In 1953, the journal Nature published back-to-back papers, James Watson and Francis Crick's DNA double helix model, and separate experimental work from Rosalind Franklin's King's College London group. The world now had the double helix. But recognition would unfold far more slowly and unevenly. Soon after, Rosalind Franklin left King's College London for Birkbeck College, turning her formidable X-ray crystallography skills toward viruses and producing groundbreaking structures there. Rosalind Franklin died in 1958, age 37. In 1962, James Watson, Francis Crick, and Maurice Wilkins received the Nobel Prize in Physiology or Medicine for DNA. Today, the story sounds different, and that's where the hope in Rosalind Franklin's legacy truly lives. Today, textbooks celebrate Rosalind Franklin, Photo 51, and her X-ray crystallography as foundational to understanding the DNA double helix, inspiring generations to pursue science with courage and integrity. Rosalind Franklin never heard the Nobel announcement, but her work echoes in every biology textbook. Thanks for watching. Subscribe to explore more untold stories that shaped our world.